And no, that's not what I want. That's what I want. Why isn't that not sharing? No. Okay. Sorry, guys. Let's try this. Okay. So what we're going to talk about today is how we apply microbiology to benefit us as humans. So the services. So some of this is going to be treating our water, treating the water that we consume and treating the water after we've consumed it. And a large part of that is using bacteria to break down the waste, the organic waste, the solids that is present in sewage. We'll talk a little bit about that. We'll talk about how the biochemical pathways of microbes and the metabolic processes that they carry out are used for the benefit of our foods and beverages, whether it's beers and wines or kombucha or things like cheese and bread. All of these things are made possible by the presence of microbes. Then we'll talk about the differences between getting sick from food that has toxins from microbes versus getting sick from foods that have infectious microbes in it. Okay. And then also using microbes to get a particular flavor, right? Not everybody likes Limburger cheese. Anytime I break out the sauerkraut, everyone else in my family like clears the room. They don't like it, right? So some of these things are used to get a particular flavor or odor, and it's from the microbes. And we'll talk a little bit about that. And then finally, how these things, these microbes are used on an industrial level to make things like enzymes, antibiotics, vaccines. And so all of these things come together to actually benefit humans. And if we didn't have these microbes, we wouldn't have any of these available things that we enjoy. Okay, so a couple different terms to discuss. Applied microbiology. That is taking microbes and using them to either treat things like wastewater or to do bioremediation, using microbes to clean up oil spills, using microbes to break down plastics. As one of your, I know you have a cut. I'm sorry.
Don't touch it.
Oh, I'm sorry. So applied microbiology, using these microbes to break down things for our benefit. Industrial microbiology is using microbes for the manufacturing of foods, medicines, different agricultural products. For instance, you can combine biotechnology and industrial microbiology in agriculture to make biopesticides, right? Using bacteria plasmids to add genes to plants that make them resistant to certain pests. And then you don't have to spray harsh chemicals on those plants. And then fermentation. Fermentation is done for a number of things, whether it's food products or to get the byproducts of those fermented microbial metabolites. Okay. So these are the things that we'll be talking about in today's lecture. And it's kind of nice. I feel like it's good to kind of switch off of the whole um, uh, nastiness of microbes and see how they're working for us in a positive way. So this is just a generic image of a typical water treatment facility. You'll notice there are all these different pools, these circles here. Those are just large um, containers of water, and they pump the water through all these different um, uh, buildings, these different facilities, to clean out things and then channel them through these different pools that are seeded with lots of bacteria that help break down, again, the organic wastes. And so these microbes can be used for purifying the water to a certain degree. And again, it's a stepwise process going from one station to the next. And each station does a different decontamination or purification or filtration process. And it usually involves taking the water and putting it in some type of reservoir where the sedimentation starts to form. All that organic matter in the wastewater settles to the bottom. Okay. And then, of course, this water does need to be treated so that you don't have certain microbes that like to grow in that. It's seeded with the microbes that break down the waste, but you can also now have things like cyanobacteria, photosynthetic bacteria that would love to be in this water because it has all kinds of nutrients in it. But those aren't being used to break it down. Those would just grow in it as a result of it just being available. And then it's pumped to different tanks, further settling. They aerate the water. So they put in water, they put in air, O2, to bubble it up and feed the aerobic bacteria so that they can do their thing. And then finally, it's usually treated with some kind of chemical, usually chlorine, and then pumped away to another location, whether it's to be used for agricultural irrigation, or in some cases, like I believe LA has a water treatment facility that reuses 100% of the water. So they take in the wastewater, they treat it, they run it through all these different holding facilities, and they pump it out, and it's drinkable. So varying degrees. In Tahoe here, once they treat the water, they pump it up over Luther Pass and then out to be used for agricultural water. When we treat sewage, there's often three phases: a primary, a secondary, and a tertiary phase. And each of these phases have different things that they're removing from the water. So the primary phase is going to remove the floating bulky things like, toilet paper that didn't break down. If you guys were to tour STPUD, which we usually do, you'll see they'll show us this primary phase, and there's all kinds of things like cigarette butts and flushable wipes. And the biggest take-home message they say from Stupid is, don't use flushable wipes because they're not flushable. Okay, it's a huge problem for them, actually. And then there'll be a lot of kind of gross things like food that doesn't get completely broken down in our systems. So there's lots of like kernels of corn and stuff like that.
Daddy!
What? Oh, okay. All of that has to be removed from the water before it goes into the next phase, the secondary phase. And that's going to be removing the organic matter, the stuff that doesn't get pulled out by the first initial filtration. It's small enough that it passes through. And this is where we're going to have a lot of the microbes do their action. Okay, so this is where we put them into those holding tanks. We put the water into the holding tanks, and then the microbes start breaking down that organic matter, like poop and stuff. And then tertiary phase, that's when you take this water that has been treated by microbes and you do one last filtration, you do a disinfection, you add bleach, and then you're ready to pump it somewhere else. Anaerobic digesters. So in combination with the aerobic microbes, you're going to have anaerobic microbes that are going to take this sludge, this sediment, and start breaking it down for their energy purposes. Okay. And so this can be done in a really high throughput manner in terms of how much water can be treated. Millions of gallons of raw sewage a day by this process. It's very high stream, high throughput. And then, like I said, once it goes through these steps, it's released. And depending on the level of decontamination, that will determine what's done with that water next. Tahoe pumps it out because they want to keep Tahoe blue, so they don't want that water that has, still has probably some degree of nutrients in there from getting back into Tahoe. And so this is an image of what that kind of looks like at a typical treatment plant. You have the primary stage here with your raw sewage, everything that gets flushed down the toilet, everything that goes into the storm drains. All of that stuff has to be separated out to where it's not going into your holding tanks, right? And then once that's filtered out, you take the water and you put it into this holding tank, and you have what's called the activated sludge. Activated meaning that it's been seeded with these microbes to start breaking down the organic waste. And then tertiary stage, you pump it through to be treated with chlorine, and then again released. So this is a typical facility. You have your water source here at number 1. And then all this is showing you, I'm not going to go through these different numbers, but it's just showing all the different stations that this water goes through for its initial decontamination and filtration to its subsequent and final chlorination to where you get to the last step where you're drinking it. Okay. So an interesting situation. This diagram is probably going to be just for water treatment, not wastewater treatment, because again, usually wastewater isn't recycled. This image on the right is an image of STPUD. This is from last year when I took the micro students on a field trip here, and you can see in the background there's a large tank here. That is the holding tank for breaking down the microbes. And then behind all of these students is me taking the picture, but then even further is LTCC. So the STPUD is right next to LTCC. And some of you, if you've been to campus before the closure, you'll notice on certain days it kind of smells funky walking to the building. That's because we are right next to this treatment plant. And then here we are walking along this bridge here, and to the right, this is being aerated, so you can see kind of the foam on top. That's just a bunch of bubbles being released to give the microbes their air and to kind of turn the sediment. And then over here, this is a tank that is not being used. It's mostly empty, but not completely empty, because they want to keep the microbes alive. So they switch between these two different tanks, but they never drain it completely because they want to keep the microbes active. And it really is a self-sustaining system. They don't need to reseed these vats with new microbes, right? Unless they have some kind of die-off event, which occasionally happens. Barring that, it's pretty self-sufficient. Any questions on that, on wastewater treatment?
Hey, Doc, I got a question. I see you guys walking the catwalk there. Were you guys wearing any masks? I mean, is there any problem with stuff coming up from all the aeration and
No,
potentially ingesting that and contaminating?
no. So the only time any PPE was worn was actually in the lab when we toured their lab where they analyzed the water. We had to wear goggles and lab coats and stuff. But no, here it's all of this stuff is contained. So the microbes and everything are living at the bottom of these pools, and they're non-infectious. They're not microbes that would infect humans, and stuff really isn't being released except for odor. So you could wear a mask if you didn't want to smell it, because it definitely isn't pleasant to smell. But in terms of harmful, byproducts or microbes, not a concern. But good question. Any other questions? Winter, yeah, how does that affect the process? Well, I asked that question, and I asked if they put covers on and stuff, and the water is warm, and so typically the snow just melts, and they have different drains on it so that it doesn't overflow. They can control the level of water in these tanks, so they really don't need, they really have these open year round. And on days of big snow, it's really only an issue of like everything else, plowing the roads in between the different facilities. The microbes are fine. They're nice and warm, and it doesn't, there might be a heater. There might be some kind of heating element too to keep the water warm to prevent die-off, but good question. Okay, so that's utilizing the power of microbes to treat our water. What about microbes in food? Microbes and humans have this competition. It's not just warfare between them wanting to infect us and us wanting to kill them off. There's competition in our food, right? You get those raspberries, you bring them home, and they weren't the ones that were irradiated, and next thing you know, you got a bunch of mold that's competing for your raspberries. They want to get those nutrients too. Okay, and so that would be something that would be a negative, but a lot of effects are beneficial, positive, right? So fermentation, right? This is using microbes to break down essentially sugars that will give us a desired flavor, a desired texture, or a desired experience, right? If it's carbonated, it's going to be fizzy, and that gives you a different, like, effervescent experience than if it was flat. But there's also a lot of bad effects that come from them wanting to use our food, and that can be in the form of foodborne illness or just ruining our food, right? If it may not make us sick, but now I can't eat those raspberries because they're all covered in mold. So these are going to be things that are undesirable, right? It's going to make your produce smell terrible. It's going to break down the nutrition, so you're not going to eat it, right? Even if it's full of bacteria that can't infect you, it's going to extinguish the qualities of that food and make it no longer appetizing and nutritious. So it, they can serve both sides of the coin when talking about their roles in food. In terms of fermentation, it's used to make bread, right? So the yeast breaks down the sugars in the flour And one byproduct is CO2, which causes the bread to rise. Fermentation of barley gives us beer. Fermentation of juices, particularly grapes, right, gives us wine. If we ferment cabbage, we have sauerkraut. If we ferment cucumbers, we have pickles, right? We can make certain vinegars by fermenting juices completely. Okay, if you take a juice and you ferment it a little bit, you'll get some acetic acid, which is vinegar. If you continue that process and exhaust all of the sugars, now you have a much higher concentration of acetic acid. And then we use that for vinegar. And then to make things like cheese and yogurt, right? And if you like cheese, you probably love cheese, right? Like people either don't do it or they just, I don't know. I don't know if anybody who is like just, I could take it or leave it, right? Cheese is fantastic. And if it weren't for microbes, we wouldn't have it, okay? With bread, right? Again, the leavening is because the flour is broken down. The sugars in the flour is broken down to release CO2. And typically that microbe that's associated with making bread is Saccharomyces cerevisiae. It's a yeast, baker's yeast. And in fact, in the COVID era, I don't know what it's like now, but for a while it was kind of hard to buy yeast at the store. There was a shortage. Everyone was stockpiling it because they thought that, you know, they'd have to make their own bread and get back to basics. But this can also serve flavor and odor. So a rye bread is going to get that characteristic rye flavor from using rye flour, ground up rye, but then also from the lactic acid from a lactobacillus bacterium and several others. Whereas sourdough is going to be also using bacteria and yeast. And, you know, sourdough has a bunch of different strains, but the San Francisco sourdough bread that is the iconic one comes from Lactobacillus San Francisco. Okay. This also helps condition the dough to make it more workable. So if you make I don't know if anybody makes sourdough bread, but I kind of took it on last year as a hobby. There are certain times where you have to let the dough rest. Okay. And so when you're doing that, you're essentially allowing the microbes to break down some of the flour sugars, and that makes it more easy to knead and shape. Okay. So lots of different services that they provide. They allow us to leaven the bread. They allow us to have different flavors for the breads, and they allow us to make the bread more workable. Beer and alcoholic beverages, I'll add kombucha to this as well. It's used by fermenting sugars in fruits or grains that produces ethanol. Okay, so you have C6H12O6, that's sugar, and that yields 2 ethanol molecules and 2 carbon dioxide molecules. Okay, and that's why beer is carbonated. Okay, it's not like soda where they add in carbonation. It's a natural process. Sparkling wines, those are sparkling because they've allowed that CO2 to build up in that whatever container it's in while it's still fermenting. And then, you know, depending on what you use as your starting material, whether it's a fruit or a grain, it's going to give you a different type of alcohol, right? And depending on how much sugar you add at the beginning, it's going to determine what the overall alcohol content is, right? If you give it more sugar later on in the process too, you give it another spike of sugar, you can drive up that alcohol content. And so humans have played around with this for a very long time, and we have lots of different variations on the same theme, basically. Does anybody know what the oldest fermented beverage that we know of is? Any guesses? What have humans been fermenting the longest?
Beer or wine is my guess.
Good guesses. And one
Or
of them,
mead or something.
mead, yes. So honey mead actually is what I heard. I would have, before I heard that little trivia tidbit, I would have thought that wine was the first one, but actually mead was, as far as archaeological evidence can suggest, was the first or one of the first beverages to be fermented. And this served a role not just because people wanted to, have the effect of alcohol. Why else was fermenting beverages so important?
Because it was safer to drink than
Yes,
contaminated water.
exactly. So before humans had these fancy water treatment facilities that we just talked about, they had no way to decontaminate. They didn't even know what it was about water that made them sick. They didn't even know that microbes existed once upon a time, but they knew that they could drink beer or wine and not get sick. But if then they went to the creek and, drank from there, they would get sick. And so that's why there's such a long history of men, women, and children drinking alcoholic beverages morning, afternoon, and evening, right? Because it was safer for them in a kind of an odd way, right? And interestingly, so I'll digress because this is a casual lecture. We're just going to take none of this is really going to be an emphasis. So the Enlightenment period coincides with the introduction of coffee and coffee houses to Europe. Because before that, all the intellectuals hung out at bars, taverns, right? Pubs, whatever you want to call them. And, and drank alcohol and then did their armchair science chats and stuff. And then all of a sudden these coffee houses showed up. And instead of getting intoxicated, people were drinking coffee. And I'm not saying that coffee, I'm a big fan of coffee, if you don't know, but I'm not saying that coffee, created the Enlightenment period, but it did facilitate outlets where people could drink non-alcoholic beverages. And I think that's more conducive to, thinking intellectually. So another little trivia tidbit for you.
Hey, Doc, I've never heard the term mead. Is that M-E-A-D?
MEADE, I think. Maybe MEAD. One of the two. But yeah,
I guess I'm asking what is mead?
Well, it's usually it's very similar to beer, only instead of using barley, they use other things like honey. So the mead I'm familiar with is like a honey-based beer. So I think they add in other components of beer. Maybe they add in hops and malt and stuff like that, but they put honey in there as the sugar source as opposed to barley. Look it up. I don't know if I've ever had mead. Has anybody had mead before? And if so, was it good? Was it bad? Was it okay? How did it compare?
It tasted really good. There's definitely different kinds. Sometimes, you can use different fruits with it, just like in wines. Sometimes they make it sparkling. Sometimes it's not, the sparkling. Different alcohol contents and the different honeys make it taste different.
Does it have a more similarity with beer beverages, or would it be more similar to wine in your?
More similar to wine.
So it's more like a wine. Okay.
Yep. Yep.
Gotcha.
I'd even say it's like a Kurt cider.
Oh, okay.
Yeah.
Yeah.
Sometimes.
Yeah, like a hard cider type thing. Interesting. Okay. So, the steps are the malting, the mashing, adding hops, then you let it ferment. And IPAs are really popular right now. IPA stands for India Pale Ale. Does anybody know why IPAs have that characteristic hoppy flavor to them?
Well, the hoppiness comes from using a certain type of hops, but IPAs tended to be stronger, have more hops and higher alcohol content, I think, so that it would preserve better when they would carry it back from India on ships, something like that.
Yeah.
I'm not really sure.
The version I heard was when British colonialists were going to India, they put a bunch of hops in there to slow down the fermentation process so that by the time they got there, they didn't exhaust all of their beer stores. So it could also have been for the return trip as well. But the idea was that adding more hops slowed down the whole process. It also led to a much stronger alcohol content at the end of the trip and a much more robust flavor. But that's why it was done. It wasn't done to achieve a certain desirable trait. It was done as a step in this brewing process. And then after, of course, you age it and stuff like that. So I think Sean Ryland once taught a special topics course all about brewing. I don't know if he'll resurrect that at some point. Maybe it wasn't here. Maybe it was when he was teaching somewhere else. But I think that'd be an interesting class. I would take that class. Winemaking, similar kind of thing, right? You got to press the grapes, whether it's now using machines or back in the day using your feet. And then there's usually some kind of treatment. And we're going to do a fermentation lab later on in the quarter, where you guys will just take some grapes and mash them up and do the same thing. They usually do a heat treatment to kill off any contaminating bugs, okay? And then re-inoculate it with the yeasts, okay? So it can be done either way, but this is a more safe way. But if you just took a grape off of a vine and didn't wash it, and you left that little kind of film on the outside, that would be enough to essentially make your own alcohol. And generally speaking, it's probably okay to skip that sterilization process, because remember, alcohol is one of the what's that we talked about last week. Germicides, right? So things aren't going to be able to grow that well in an alcoholic environment, especially wine, which has a higher alcohol content than beers. But I think that a lot of companies, a lot of wineries are going to do that heat sterilization just because it's safer that way. You're selling it, so you don't want to be liable for outbreaks. And then once you do that, you put it in a barrel and you let it ferment, and then you let it age, and then you filter off all of the particulate and you bottle it up. But those microbes are an integral step. No microbes, no fermentation, no wine. Okay. That's alcohol. What about milk? Milk is a really great medium for culturing bacteria. Think about all the different things we use dairy for. Cheese, certain butters. So yes, regular butter is just churning it and getting the cream to rise, but things like buttermilk use bacteria. Kefir and kumis. I've never heard of kumis until I started teaching microbiology, but it's a fermented milk from usually from horses and donkeys, which I mean, I guess that makes sense that some people do that. I've never tried horse milk before. But then think about all the different cheeses we have, all the different varieties, whether it's soft cheese or semi-soft or hard cheese. All of these have their different qualities based on the microbes that are used to inoculate that milk, right? So things like blue cheese and gorgonzola are going to be using a mold, this penicillium, not the same penicillium species that's used for penicillin. This is a different penicillium, but that's using mold. Swiss cheese has all the holes, right? Well, if you bought a block of Swiss cheese, you'll know that those aren't actually holes. They're just like pockets. There's, inside the cheese. And when you slice it, of course, it looks like a hole. But that's because they're using this propionibacterium that ferments the sugars in the milk. And then one of the byproducts is carbon dioxide. And some of that carbon dioxide cannot, because the fermented milk is dense enough, it can't break through that milk to get out. And so it forms these pockets. Yogurt, lactobacillus, right? And some of these things like yogurt has different cultures that are claimed to be beneficial, probiotic for us. And I think I've already mentioned, that the science behind that maybe suggests otherwise. And when we talk about the microbiome more, we'll get into that. But nonetheless, yogurt's still pretty good for you. What time is it? We got about 10 minutes left. So now let's talk about the bad aspects of microbes and their roles in food. Food poisoning, right? Some of you guys are, I think, doing your proposals on some level of food-borne illness, right? And there are two types. You can have food intoxication, meaning you get sick from eating something that has been contaminated with bacteria or some microbe, but usually it's bacteria, and those are secreting toxins. Remember, we talked about endotoxins and exotoxins a little bit. And these exotoxins they secrete, some of them can be just metabolites. Some of them are secreted to kill off other microbes, right? To stake out their territory. And these can be intoxicating to us as well. And then there's food infection. And that's when you actually have food that is contaminated with microbes that are still active and alive and able to infect us. Okay, so think salmonella. So two different ways to get sick from microbes in food. And here are some tables to kind of outline this. So food intoxications can come from things like Staph enteritis, and that's going to be from the Staph aureus bacterium. Oftentimes in things like custards and cream-filled donuts, hams, pretty common. Most foodborne illnesses come on rapidly, and that's how you know that you got sick, right? If you seem to just all of a sudden need to head to the bathroom, then chances are it's a foodborne illness. And the nice thing is, because they come on quick, you're usually able to process that intoxication rapidly as well. So it's not a chronic, prolonged infliction. And if you want to know more about that, chapter 18 talks about Staph aureus. Botulism, that is what's used for people's faces. They inject this toxin into their skin, but if you were to consume it down your digestive tract, it could make you sick. If you don't properly can your foods at home, like we went to Oregon and picked a bunch of blackberries over the summer, and then we made a bunch of jam. And I was planning to give some out as presents. I'm hoping that I did that process correctly. So far, we haven't got sick, so fingers crossed. And then Bacillus cereus, or B cereus. You need to be serious about Bacillus cereus because this can be found in things like reheated rice, right? So you make your rice and you don't eat it all and you put it in the fridge and it's got all those nice starchy sugars that bacteria love. And then you reheat it, but you don't reheat it enough to kill off everything. And then bam, you're sick. So those are some of the more common toxins. There's a lot more food infections than food intoxications. Campylobacter, salmonella, from mostly animal products, right? So milk, eggs, meats, things like that. And again, if you want to read more on that, chapters 21 and 20 are good. We'll cover some of this stuff later when we talk about the medically relevant bacteria. Clostridium, shigellosis, Vibrio. Again, most of these things are going to come from inadequately prepared animal products. So if you're a raw vegan, chances are you won't have to deal with a lot of this stuff. However, E. coli is one of the few ones that does contaminate things like produce. Why? Because E. coli is in the soil and plants grow from the ground, and so they're not properly washed. Then that's where we get these outbreaks, and it's usually in prepackaged vegetables, right? So that's because it's all contained in this hermetically sealed bag, and the conditions are nice. Sometimes you get a little bit of a greenhouse effect during transportation if it's not refrigerated, where that bag is nice and warm, and so the E. coli thrive and make you sick. Any questions on this? Anybody had any foodborne illness recently? I know that's not to be a HIPAA violation. You don't have to tell us if you have, but if anyone has an experience with a foodborne illness they want to share, by all means, now is the time to do it.
Yeah, in fact, just recently, both myself and my dog had issues.
Oh, yeah?
Yep. So usually
Are
I'm
you
pretty
eating
good
your
at,
dog's food, or is your dog eating your food?
so I give her the little remnants off my dinner
Yeah.
plate, it's her one treat. And I don't know, about for 4 nights, both of us, 4 days, 4 nights, we both had it.
Wow, that's a long time.
Yeah,
That's awful.
I know. So, but we're both better. I didn't need meds, but she did.
Oh, wow. I'm sorry.
Yeah, they gave
Now,
her meds because they said that it was probably affecting her intestines and the lining, and it could actually get worse more often with dogs. So, but yeah,
do you recall what it was? Was it meat, or was it something that you prepared? Like, not to say that you did this, but like, was it something
well,
that you
yeah,
bought? Was it takeout? Was it vegetables, or like, what do you have an idea of what it could have been?
it was meat remnants. That's typically what I'll give her. Maybe a little bit of rice or potato as
Yeah.
well.
Yeah.
But that's, yeah. So I don't know, typically I eat fish or chicken.
Mhm.
So something in there, I don't know exactly what, but
Yeah.
yeah, that's,
Well, take a look at this table and maybe you can self-diagnose a little bit.
yeah.
Okay. Well, thank you for sharing, Christine. I'm sorry that happened. I'm sorry it was 4 days and 4 nights. That's pretty gnarly. But
Well, it was the roughest for the overnights because she had to go out like at least twice an hour.
oh
It was, but then she'd be like weak all day.
yeah.
Like, so all better now though.
No bueno. Well, good. I'm glad. Yeah, my dog definitely got into something a few months ago, and it was evident in the middle of the night. So, yep, that happens. And some of these things, interestingly, we've talked about how microbes have certain species tropisms and stuff, but that's not always the case. There are certainly microbes that just infect mammals at large, right? And so the same things, like your dog isn't going to get influenza from you. Now there have been some cases where I think there was a pet tiger at a zoo, or not a pet tiger, but a zoo tiger, not a Tiger King tiger, but that had COVID. And there have been some veterinary reports, but definitely there are these foodborne illnesses that can infect both humans and our pets. Okay. So how do we prevent this from happening? Well, one thing, no, I'm almost done, buddy. One thing is that when we're preparing foods, we do aseptic technique. What is aseptic technique? Well, you guys haven't been able to really experience aseptic technique because we haven't been in the lab. But what that means is that is just decontaminating. Hey, go. Decontaminating. Hey, decontaminating your hands, the tools that you use, the containers for the food, right? So aseptic technique in surgery is, of course, to scrub, take off your jewelry, get under your fingers, and then once you've scrubbed your hands, you have somebody help you put on your gloves and stuff. It doesn't mean that you go, okay, and then start filling your jar with jam, right? Aseptic technique. You have to sterilize all of your equipment. You can't sterilize your hands. You disinfect your hands, right? With antiseptics. And just doing that goes a long way, right? I've been making kombucha for like 4 years now, and I've never had my jar get mold or contamination. I've never had an issue of people getting sick drinking it. And I ascribe a lot of that to just being a microbiologist and having this aseptic technique burned into my habits. And so I take that very, very seriously when I'm handling that. And then of course there are other things to limit food poisoning. One is to heat. Autoclave, of course, is going to really destroy a lot of the nutrients in food, so that's not used very often. But pasteurization is. Cooking your food is. You don't want to eat a chicken breast rare, right? Because you have a potential there for foodborne illness. Why is it okay to have steaks rare, but it's not a good idea to have a hamburger rare? Because steak has contamination usually on the outside of it, but inside it should be mostly sterile. Whereas with ground beef, you're mixing it all up. You're grinding it up and it's all just mixed in there. So it's better to cook that through. Cold, right? So what's that? That's not microbicidal. It's microbiostatic, right? It's just keeping things at bay. Radiation, we've talked about that. Chemical preservatives like salt, it dries out the bacteria. It doesn't force the water in, as I butchered a few weeks ago. Drying it out, desiccation. Spraying of bacteriophages. So actually putting viruses into the food to kill off the bacteria. Okay. And so that's a good place to stop. We'll go into industrial microbiology on Wednesday. Any questions on that? That was, hopefully not, maybe that was kind of a breath of fresh air from the pace that we've been taking more recently. Any comments, concerns about the lecture or about anything really in class?
I got one for you, Doc. We were carrying a sugar Halloween hangover, so I hiked my boys yesterday up to Lake Cathedral, and inside that lake at higher elevation, there's these little green kind of fluorescent globules that clung to the rock. And just for the fun of it, we scooped it out of the cold water, and it was real slimy. And again, it was see-through, and it kind of dripped through the fingers. What is that? and should my boys be playing with that?
My guess is that it's some kind of algae or cyanobacteria. I don't know. I've never been to Cathedral Lake. I'd have to check that out. But did you get a picture of it?
No, I could probably pull something up from the web and share, but it was something I haven't really seen or touched before. So it's kind of.
I mean, unless they were slurping it down, I'm sure it's fine to just touch on the outside. But that's interesting. My guess would be some kind of algae.
Yeah, really tubular. Up to like probably 6 inches long, diameter of like maybe an inch, and just fun tactilely to play with, right? To touch.
Yeah. No, interesting. I'll have to check that out. Thank you for sharing that. Anything else, guys? Okay. Well, remember, your draft is due this Friday. And so if you would like to chat on that, I'm happy to hang out for a while since our lab has been postponed. And we can brainstorm, or I can give you some helpful pointers if you're still, I don't want to say struggling, but just still running into some roadblocks. We can use Wednesday as well. So there'll be a labster, but we can use the lab period also to work on proposal ideas and stuff. Now, if you aren't, if you're okay and you're good on your own, then I would recommend that you use this period, next couple hours, to work on your proposal, because remember, the draft is due. So look at the template and fill in those different gaps. Okay. And so like I said, if you maybe we'll take let's take a 5-minute break, and I'll come back at 11. And if any of you guys are still on and want to talk, then great. And if you're feeling strong and confident and you don't, you're free to go. Okay. So we'll leave it at that. And for those of you I don't see at 11, I will see you Wednesday. Okay.